All right, everybody, we're going to go ahead and get started with our cells, biochemistry and cells review. I made a little PowerPoint. I'll share it with everybody. Let me get my picture out of the way here. And if my picture covers up some things, my apologies. All right, so let's go ahead and start talking. So the first thing, I went entirely based on your learning targets. So I looked through the college learning targets and I just tried to make sure that I pulled things that you had seen before that came right out of our learning targets. So the first thing that it mentions in the learning targets for biochemistry is your need to know about the special properties of water. Remember that water is a polar molecule and that water forms hydrogen bonds. And are hydrogen bonds strong or weak bonds? Super weak bonds. They break really easily, but it makes incredibly important characteristics. You might remember the terms cohesion and adhesion. Does anybody remember what those are and the difference between co and adhesion?
Cohesion sticks to like the same molecules, and adhesion sticks to other.
Cohesion is the same molecule, so that would be water molecules sticking together. Adhesion is sticking to something different. I always thought of like co-workers somehow being the same. So cohesion is when it sticks to things that are the same. So that's when you see something like water surface tension. The water molecules are attracted to each other. Adhesion is what allows it to move up like a capillary tube or allows it to move up some kind of a small. You see adhesion in the lab when we look at the meniscus on a graduated cylinder. You see adhesion is when it starts to creep up the walls of the glass. That's adhesion. It also has high surface tension, capillarity. That's what allows it to move up the tube. High specific heat means because of the hydrogen bonds, you have to add tons and tons of energy for the temperature to change. So the temperature doesn't change very quickly. When it finally does change, there's evaporative cooling. That means when it evaporates, it makes your body feel cooler, or whatever the surface is, the temperature goes down. Ice floats because of hydrogen bonds. When it forms a solid, usually solids are more dense and so they sink. But in this case, the hydrogen bonds cause the water molecules to be far apart. And so the ice floats. And then it's a universal solvent. So that it can dissolve lots and lots of different things. Whoops, I went too far on one of these, trying to do two computers at the same time. And then it also has, in this case, it is where acids and bases come from. Remember that water can break apart. It's H2O, which means it's an H and an OH, and they can separate from each other. I will say that we're going to have a unit on acids and bases, and that will become clearer as we go. So don't worry about acids and bases right now. We will have a unit in chemistry about acids and bases before your test, and so you'll be good to go on that. All right, and then the other thing that I just wanted to remind you quickly, why cohesion, one really big reason cohesion and adhesion are super important, has to do with transpiration. Remember that trees don't have a heart. So how are they pumping tons and tons of water from their roots up to the top? Part of that is powered by the sun. Water is evaporating, but then the water molecule, each water molecule pulls on the next water molecule. That's cohesion. And they're attracted to the tubes in the tree itself, and that's adhesion. And that allows the water to move upward. It's a very crazy concept, but that's because of water. Remember that without water and its wacky properties, there is no life on this planet. It is totally dependent on water molecules. All right, the next thing we're going to talk about are the four main kinds of macromolecules. The carbs, the lipids, the proteins, and the nucleic acids. Just shout outs quickly. What are the carbohydrates? What are they? Okay, mono and di and polysaccharides. So examples would be like sugars and starches. And then the lipids. What are lipids? Those are the fats. This is very confusing for me to be doing two at the same time. Good. They're the fats and the oils. That's correct. And then proteins. You don't need to be able to name a specific protein, but what is a type of protein that's wickedly important? They do things. Nope. They do things in the body and they speed reactions. Enzymes. Yes. Enzymes are a type of protein, but they're responsible for all of our life functions, and they speed up all the reactions in our lives. We'll go in depth about enzymes when we talk about cell energetics later. So we're not going to talk a lot about enzymes right now. And then the last category are the nucleic acids. And what are we talking about with nucleic acids? DNA and RNA. And again, we had an entire unit on that. So we just touch on it today. We don't go into a whole lot of detail on that. All right, so then they also wanted us to talk about polymers and monomers. Remember that polymers are what these molecules are made of. They're made of a bunch of monomers put together to make a polymer. The one exception to that is lipids. Lipids are not made of polymers, but all the other, of monomers, but all the other macromolecules have a bunch of simple molecules that come together to build a more complex molecule. So the monomer is the simplest unit and the polymer is the more complex unit. And they come together with dehydration synthesis, or they can be pulled apart with hydrolysis, which we will review in just a moment. All right, so let's just practice the monomers and the polymers. For the carbohydrates, the monomers are the monosaccharides. And then carbohydrates can be straight or branched. I just added this image. It's not working. What am I screwing up? Oh, it's right there. Okay. I added this image. I know it's totally hard to see, but these are examples of carbohydrates. These are glucose molecules. And they're either branched. It says moderately branched, that's starch. Highly branched is glycogen, and then not branched at all is cellulose. So for some reason they don't need you to know cellulose, starch, and glycogen, but just that it can be branched or that it can come in one long chain. So just be aware of that. Good. Okay, the next polymer that I wanted to talk about are the proteins.
Wait, I do have a question. So
question on the carbs.
Does that mean like it's like a unsaturated fat?
We'll get to that. So no, it's not like that, but we'll get to it.
How it's like
so the unsaturated fats have a bend in them, but they're not necessarily branched. Branched means that there's actually it's going off in a different direction, so there's more than one. So it makes a Y, for example, as opposed to just a bend in it, which would be what happens with the lipids. All right, for the proteins, they're the polymers. What are the monomers of proteins? Amino acids. That's what they're built of. And remember that there's about 20 amino acids. We'll talk a little bit about the amino acids in just a second. Okay, and then, and the other thing I do want to mention about the amino acids, if you just take a look, try to locate the amino acid that's up here. I thought I had an amino acid. Oh yeah, I have no. I do have an amino acid. It surprised me. This one is the amino acid right here. So the amino acid has an amine group, the NH2 group right here. This is the carboxyl group over here. And then coming down from the top, so for those of you that are online, it's this one right here. This is the amine group, the carboxyl group, and then down here is the part of every amino acid that's different. And we call that the R group. And the R groups are wickedly important, and we're going to talk about the R groups more in a few minutes. But they're all a little bit different. Any questions about the amino acid? There's 20 of them, so that means there's 20 R groups. Okay, I'm moving on. The polymer then for the nucleic acids, the polymer is DNA or RNA, and then the monomer would be the nucleotides themselves. The nucleotides are made of a phosphate group, a sugar, and then one of the nitrogen bases. So for those of you online, you can see the phosphate group there, the sugar is there, and then this is one of the nitrogen bases. Phosphate, sugar, nitrogen base. Remember that the nitrogen bases in DNA, A, T, C, G, and then RNA has its own nitrogen base, U. Again, you just need to be aware of them in this unit. Coming up, we'll talk about how they're opposite of each other. We'll talk a lot more about the nitrogen bases, but right now you were just being introduced to them. All right, let's talk about dehydration synthesis. Does anybody remember a little bit about, does that sound like a familiar thing?
So lipids don't have monomers?
Correct. There's no building block. The question was lipids don't have monomers, and that's correct. There's no building block of a lipid. A lipid is a long chain of carbons and hydrogens, and we'll see it in just a minute to review it because it'll be hard to remember that.
What's going on with the glycerol or the glycogen or something?
Yeah, we'll take a look at that in just a few minutes. Okay, so dehydration, to synthesize something means to build it. So you're adding complexity here. And so this is when we remove, dehydration synthesis removes water. So we pull water out and we put two molecules together. So we say that that's anabolic, which is when we're building complexity. Endergonic means you have to add energy. I don't think I would worry too much about those terms, but they are on the learning targets. What's the opposite of anabolic? Who remembers this at all? Catabolic. And what was the really dumb way I helped you remember this? Cats breakdancing. So catabolic reactions break things apart. So if you can remember that one, then anabolic is the opposite. Anabolic is putting things together, is building in complexity. So this one is anabolic. We're building in complexity. All right, and all of these reactions require enzymes. Every single reaction in your body, it can happen naturally. It can happen without an enzyme, but it would happen incredibly slowly. So your body has these enzymes to make these go a lot faster. We'll talk more about enzymes a little bit later. And then hydrolysis is the exact opposite. To lyse something means to break it up. So this would be adding water in this case, hence the prefix hydro, and it's catabolic, which means you're breaking it apart. So this is the equation that's on the other below it. And it releases energy. And it requires enzymes also. My picture's in the way on the video, but folks can look at it online. Okay, I said that I would talk about lipids specifically, and that's because they don't have monomers. So I just want to mention a few things. Remember the important things about lipids, like they're nonpolar. So that's a unique aspect of the lipids. They don't dissolve well in water. Remember the difference between saturated and unsaturated. So unsaturated have a double bond in them, and that's what Tara was asking about. That causes them to have a kink. And at every double bond, there will be a kink. Keep in mind, and this diagram I like, the red up at the front, the red, there is a double bond there. That bond is always there. That's part of the carboxyl acid group. It always is there, and so you're not paying attention to that. What we're looking at is the single bonds in what we call the hydrocarbon chain. And if it's all single bonds, we call it saturated. If it has one single bond, it is unsaturated. And they have fancy names for it, like monounsaturated, diunsaturated, polyunsaturated.
I saw that in the
You don't need to know it.
AP book, and I was like, what is that?
Yeah, it's telling you how many double bonds there are. So polyunsaturated would have 3 or more in that circumstance. Okay, and then remember phospholipids. Phospholipids are super important in terms of biology because they're found in the cell membrane. That's what the lipid bilayer is made of. And they're a little bit different than regular lipids because they have a polar head. And remember, the polar head is attracted to water. That's what this diagram down here is. So we often draw it like a head with 2 tails coming down. And that's the phospholipid. This is the more, detailed structure of it. Here's the lipid. The pink is the polar head. And then they make a bilayer for the cell. And that's what the cell membranes are made of. Okay, and then the last type to just be aware of the lipids were the fused rings. They look a lot like what? What could trick you? Like if you were identifying these as a macromolecule, what do they look like? They look a lot like the sugars. So the difference is that they have lots and lots less oxygen. So remember that the carbohydrates, just one example, glucose is C6H12O6. So it's got the same amount of carbon and oxygen. The lipids have hardly any oxygen in them at all. So they have usually 2 oxygen molecules. You can see the 2 oxygen molecules up at the top here. That's where the two oxygens are. But then they don't really have much other oxygen to speak of. So that's part of how you're going to identify them. All right, next we'll move on to proteins. Obviously, proteins are wickedly important. Proteins are long chains of amino acids joined by, this may be a little bit harder,
Something?
peptide bonds. There we go, peptide bonds. We haven't heard that word in a while.
Which one's phosphodiester or whatever? That's nucleic acid, right?
Yes, that's correct. All right, and then the R groups, remember the R group is what's different between each amino acid. So I've got a general amino acid here. There's the amine group, here's the carboxyl acid group, and here's the R group. There's 20 amino acids, so there are 20 R groups. The R groups are wickedly important because they give each amino acid a different flavor, a different characteristic. So some of them are polar, which means they have a slight positive or negative charge. Some of them are fully ionic, which means they're actually missing an electron or they have an extra electron. So that means they're positively or negatively charged fully. Some of them are non-polar, so they're afraid of water and they don't want to be near the water, so they tuck into the inside of the amino acid, of the protein. And then remember that proteins have primary, secondary, tertiary, and quaternary structure. So the primary structure is the chain of amino acids, and that's what's determined by our DNA. So our DNA tells us, remember, 3 letters at a time, it tells us which amino acid. And when we mutate one of the letters of the DNA, then we might get a different amino acid. And that would change the primary structure. Now, if it's a similar R group, if we change amino acids and it goes from being polar to polar, no big deal. But if it's a different kind of amino acid, it goes from being polar to non-polar, then you're going to totally change the shape of the protein and the protein will not function correctly. So it just depends what the change in the DNA. And we'll learn more about that. We'll review that again coming up.
So wait,
So secondary structure, that's what's this picture, my image is on it on the online right now, but secondary was the alpha helices and the beta pleated sheets. That's a hard one to remember. That's just due to hydrogen bonding. So that has nothing to do, that's hydrogen bonding right here. That's what those dotted lines are representing. And it's not about the R groups. It has nothing to do with the R groups. That's just about the backbone itself, hydrogen bonding. And it creates these curls or these kind of flat areas, as opposed to tertiary structure. That's what we did, and I have them sitting on my desk right now, but I'm not going to grab them right now. But that's what we did, the tubers. Do you remember we had the tubers and we put tacks in the tubers, and those were the types of R groups. And then we folded them and we put the polar R groups near each other, and we made the hydrophobic R groups fold away from water, and the ionic ones attracted, if it was positive, then it was attracted to a negative. So that determines how the whole protein folds. And then the last type of structure is called quaternary structure, and that's this pink and blue diagram down at the bottom. When you have more than one chain of amino acids, they all kind of glob together and you see the same interactions. There's R group interactions, there's hydrogen bonding, there's all the same interactions, but it's between more than one chain of amino acids. So they're showing 4 chains of amino acids in that diagram. Yes. Yeah. So a protein might be a whole bunch, not a whole bunch, but several amino acid chains put together. All right, moving on. I pulled this directly. These are direct quotes from your learning targets because they're kind of complicated. It's not that they're hard to understand, but they're hard for me to word differently. So it says, organisms must exchange matter. That means they take the molecules that they eat or absorb and rearrange them with the environment to grow, reproduce, and maintain organization. So when I'm a vegetarian, I need protein and I need amino acids. So I take in protein from beans or cheeses or whatever. And my body pulls those amino acids apart, rearranges them so that I can build the proteins that I need. By the way, you build most of your own amino acids. You get some of them from your diet. Okay, atoms and molecules from the environment are called precursors. And that was a hard question on one of our FRQs for this unit. So the atoms and molecules that are the small things that build to make our macromolecules, those are the precursors. So carbon, what is carbon in? Shout it out. What of our macromolecules is carbon in? Everything, okay? Carbs, proteins, lipids, and nucleic acids. Carbon is used in storage compounds and in cell formation in all organisms. What's nitrogen in? Not everything. Nucleic acids and amino acids / proteins. That's correct. Good. And then phosphorus. Okay, phospholipids. Yep, phospholipids. That's correct. And then also in the nucleic acids. So the nucleotides have a phosphate, a sugar, and a nitrogen base. So that's correct. Good. All right, we're done with biochemistry. I'm having us move on to the cell unit now. I'm being asked to go back one slide. Give me a second. There we go. Good. Okay.
Oh wait, I got it.
Okay, so we're going to talk, just reminding you quickly about the difference between prokaryotic and eukaryotic. You are made of eukaryotic cells, and what makes them unique is that they have a nucleus and membrane-bound organelles. They are way bigger and super, super complex. And that's going to become important in just a second. We'll talk about something that makes them complex. So the prokaryotic diagram here is not at all realistic. Prokaryotic cells are, I don't have a number, but this big compared to eukaryotic cells. So prokaryotes are the bacteria. And then just a reminder about endosymbiosis. Who remembers what, it says it right there, shoot. The organelles that moved into larger cells and then became our organelles. So they were bacteria. I'm sorry, I said that funny. The bacteria that moved into larger cells that then became some of our organelles. And that's primarily the mitochondria and the chloroplasts are the ones we really want to be aware of. Evidence that supports that. I think I might have that actually on the next page. Did I? Nope, I don't have that on the next page, but there's lots of evidence that supports that idea. They reproduce on their own. They don't reproduce with our cells. They have DNA. They make their own ATP. Lots and lots of stuff that supports that idea. All right, and then this also comes straight from our learning targets. Why are membrane-bound organelles important? And I'm not going to read you everything, but just get to the point. It allows cells to specialize, which is how we allow a multicellular organism to exist. Remember that you have eyeball cells, and you have liver cells, and you have big toe cells. They're all a little bit different, and the organelles allow it to specialize. Membranes isolate different areas of the cell so that special reactions can happen just in this area and not in this area. And then membranes provide surface area for more reactions to occur. So that increases, remember, it's all about surface area to volume, and that'll come up in just a second. But the more folds an organelle has, or even on the surface of an organelle, that allows more reactions to occur on that surface. All right, I am moving on. This is just a quick review of the cell structures. So I'm going to zip through it. I'm not going to spend a lot of time, but cytoplasm, I often call the juice of the cell. That's the liquid. If you squished apple cells, you would drink the apple juice. That's cytoplasm that you're drinking. Cell walls, remember that animal cells are soft and squishy because we only have a cell membrane. We have a skeleton, not an exoskeleton, that allows us to stand upright. Plants have a cell wall made of cellulose. Bacteria have a cell wall made of peptidoglycan, and fungi have a cell wall made of chitin. I can't remember if those words are on your learning targets, but I have a feeling that they are. Just to be, I don't think you would ever need to be able to say that bacteria is made of peptidoglycan. I don't mean that at all. But when you come across it in a question where they mention peptidoglycan, don't be freaked out by that word. It's what the bacteria's cell wall is made of. You don't need to come up with that word yourselves. I would be very surprised if you had to write it down. All right, and then ribosomes, we'll come back to ribosomes when we talk in our DNA unit about how proteins are built, but that's where proteins are built. And they look like little dots, and they're on the rough ER, or they're free. They're floating around in the cytoplasm. All cells, bacteria and eukaryotes, prokaryotes and eukaryotes, have ribosomes. So that's an indicator of common ancestry. So they use that as evidence of common ancestry. Think about how else you would address that. Everybody uses DNA. That would be another indicator of common ancestry. There's lots of different things that we could say that would indicate common ancestry. And then the rough and the smooth ER. The rough ER is what has the ribosomes, and it additionally compartmentalizes the cell. And the smooth ER detoxifies, oops, that's supposed to be in a comma. It makes lipids. I think that's a hard one to remember. I think that's a really hard one to remember. Okay, I'm going to move ahead. I see lots of people. People are writing lots of things down, so I'll try to move quickly. Shout at me if I'm going too fast. You can always come back to this. I will share this presentation, so you can come back and look at it if you'd like to. All right, additional cell structures. The Golgi, remember, it's that stack of pancakes. Do they have it on the diagram here? Yeah, the Golgi is right here. It's that stack of pancakes. And then it has big blobs coming off of it. Those blobs are called vesicles. And so the Golgi's job is to modify products, which are usually proteins, but there's other things. And then they package them into vesicles. Do you guys remember the walking guy?
Yeah.
Okay, the walking guy is carrying a vesicle, and it can be carried out of the cell or across the cell to another part of the cell.
I saw TikTok about a little once.
Oh, yay!
For a while, I was on like some AP bio, like
I
for you points.
I saw that on my green.
And?
Everyone's like, I hate AP bio.
Oh, no.
I honestly miss it. I prefer to chemistry.
Oh, that's very interesting.
Unpopular opinion, but I like biology better.
I'm a huge fan of life. Hey, you guys, I have to say this. I know I say it frequently, but AP biology is really, really, really hard, and it's not what anybody who likes biology wants to go into. Like you want to go into genetics, or you want to study animals, or you want to study the environment, or you want to study medicine. Nobody wants to study just what's in AP biology. So don't throw your ideas or your dreams for biology out the window based on this class. This is the foundation for everything that's really cool. Cancer research, disease research. You could be somebody who helps us get over coronavirus, whatever. There's just so much that we can do. This just introduces you to it. Okay, quickly going over this. Lysosome. In 7th grade, we called it the stomach of the cell. It has enzymes that break down cell parts, and it does apoptosis. Maybe you remember that image of a hand forming from an embryo, and our hands are webbed. And apoptosis in the lysosomes causes the webbing to go away, so that when we're finally born, we're born with fingers instead of webbed hands. Mitochondria, we're going to have an entire unit talking about this. So cell respiration and photosynthesis. But just remember, mighty mitochondria, it's going to make your ATP, and the chloroplasts are going to make your oxygen and glucose. Both of them, this is for this unit, both of them have very folded membranes that allow more reactions to occur on those membranes. That's why they're highly folded. All right, and then this is just a reminder about all the evidence for endosymbiosis. I thought I had some notes about that, but these are just things that are seen as evidence that these guys were in fact on their own at one time and have now moved into cells. Okay, cell membrane, just a reminder, the lipid bilayer, and it has all kinds of things embedded in it. There's proteins, and we're going to talk really quickly about what some of those proteins do. Glycoproteins, carbohydrates. We're going to talk about carbohydrates that are embedded, what they do. And then the function of the cell membrane is to control what's coming and going in the cell. And so we got to remind you of a few words about the cell membrane. So super important, the cell membrane, we think of it as just like a line around a cell. And when you look at it really, really close, you go, oh my gosh, it's incredibly complicated. So the fluid mosaic model refers to the fact that it's not frozen. It's constantly moving around. It sloshes around. It's a phospholipid bilayer, and we call it amphipathic, which means that the head is polar and the tail is nonpolar. So like an amphibian can move between land and water, amphipathic means that it's hydrophilic and hydrophobic in one molecule. And then it's selectively permeable, which usually means that small nonpolar molecules can pass across it. So examples, nitrogen, oxygen, and carbon dioxide. So as you're breathing in and breathing out, carbon dioxide is just passing across your cell membranes without any help. It doesn't require any ATP. We call that passive transport. We'll get to that in just a second. Okay, I'm moving on. Here's that mention I said of cholesterol. I'm not going to go into a lot of detail, but just remember that cholesterol can be embedded within the phospholipid bilayer itself, and it can cause the membrane to be more fluid or less fluid depending on the temperature. And so it helps make sure that it allows species to exist at a variety of temperatures without our cells liquefying or without our cells solidifying. Like bacon grease solidifies after it cools down. That's what would happen theoretically to our cells if they got cool enough. But the cholesterol helps our cell membranes stay viscous, stay moving. All right, and then remember that there are these proteins. We always drew them in purple in our textbook. So they are embedded in the cell membrane, and the way that they stay put is that part of the outside is polar and part of the outside of each protein is nonpolar. So the polar parts are attracted to the heads of the cell membrane, and the nonpolar parts are attracted to the tails of the phospholipid bilayer. And so they stay where they belong. And then they have lots and lots of functions. And this is a little bit introducing us into cell communication. Remember, a ligand will touch a protein and then will transfer a message into the cell. That's a different unit, so we'll review that later. All right, cell size. Remember that if a cell were to get big, it'll have big surface area. I'm going to get my picture out of the way for this group right now. It'll have a big surface area. So if you look at it here on this diagram, but the surface area to volume ratio is not very good. It's got a low surface area to volume ratio, and we want a high surface area to volume ratio. You will probably have to do a calculation on that, where you would, they give you on your equation sheet, they give you all the equations for surface area and all the equations for volume, and then you would probably be, told to say which one is going to be a more efficient cell, and you would want to choose the one that has the higher surface area to volume ratio. So why? It allows more efficient exchange of materials with the environment. The folds increase the surface area. And then as organisms increase in size, their surface area to volume ratio decreases. So elephants have a lower surface area to volume ratio than mice do. So their rate of heat exchange is very different. Elephants take a long time to heat up and a long time to cool down. And mice, their body temperatures change super, super fast because they're small. All right, I am moving on. The endomembrane system, I'm not going to say anything more about it. Just in case you see this word, you're not going to be freaked out. It's basically the idea that in the nucleus, there's instructions for building a protein. The mRNA leaves the nucleus and it goes to a ribosome. The ribosome is where the protein gets built. And then the protein gets, that might happen in the rough endoplasmic reticulum. And then in the smooth ER, it's packaged. And then it's carried to the Golgi and it's modified and then packaged in the Golgi. And then it's sent out of the cell. So that's the whole system kind of working together. You don't need to know a lot more than that. All right, let's talk about passive and active transport. Passive transport is the way things normally go. If I spray perfume over here, it's very concentrated. It automatically, because molecules are in motion, will spread throughout the room. It will go from high concentration to low concentration until it reaches equilibrium. That's passive. No energy is required, no ATP, and it includes diffusion and osmosis. However, water, I just want you to be aware of this. Water is small and polar, and water can pass through the cell membrane passively, but it has to use proteins that are called aquaporins. They're basically like hallways that the water can pass through. So that's passive transport, even though they're polar. They're small and they have their own special proteins that allows them to pass through. In the first diagram on the left, small nonpolar molecules are passing between the phospholipids, and that would be examples like oxygen and carbon dioxide. And then in the second diagram, a channel protein, which could be an aquaporin, for example, allows ions or other polar molecules to cross the membrane. And that would be an example, whoops, what just happened there? That would be an example of facilitated diffusion. If it goes through a protein but no ATP is used, that's facilitated diffusion. All right, and then tonicity. Do you guys remember this? Isotonic is what we want. We want our blood to be isotonic. Water will enter and water will leave at an equal rate, and so our blood cells stay the same size. Plants, however, they like hypertonic, hypo. They like hypotonic solutions like distilled water that allow the water to rush in, and that increases the turgor pressure, and that allows the plants to stand upright, and so that allows them to photosynthesize well. For both of us, plants and animals, salt water is bad. That pulls water out of our cells and causes our cells to shrink and causes our cells to not function correctly. Okay, moving on. I'm not sure. So you need to go. So I pulled together, how about this? Because we're getting late, I did add the water potential information, and there are some practice problems that we don't have time to do. You could skip this if you wanted to, but I'm pretty sure there's going to be a water potential problem. Not a lot of problems. It won't be worth 1,000,000 points, but just maybe take the time to refresh your memory. So I'm going to zip through these really quickly. And that is on your equation sheet. That's correct. So I'm going to be done with water potential. We've got people who need to go, which is totally fine. I started a few minutes late anyway. So when you need to go, you go. The last thing, I think maybe we're near the end. I don't remember. So active transport is what happens when we use energy. So you use your ATP, and there's two circumstances. It's moving the wrong way. So imagine if I sprayed perfume over here and it spreads across the room. That happens naturally. What if I wanted the perfume to actually be concentrated in the corner of the room? I would have to run around a room fanning the molecules to try to get them back over here. I would have to use ATP. I would have to use energy. That would be very active transport. Okay, so going the wrong direction requires ATP, and large molecules require ATP. So that includes like endocytosis, where the entire cell membrane pinches in when it's eating, or exocytosis, when we form a vesicle and then release it out of the cell. both of those are active transport because energy, ATP, is always used. Look for the diagram and look for it to say ATP, and that's your indicator. Okay, that's active transport, and pay attention to that. What did I do? Did I put two of this? Oh, holy cow, you guys. That's it. That was the last slide. I did pretty good, I think. I got you right to 3:30. So if anybody wants to stay, if anybody has questions, I'm happy to stay here, but you are all dismissed. Everybody at home, have a great day.
That was a lot of.
I didn't listen.